Hello and welcome to our water potential video. I'm Tam and I'm going to be taking today's session. So here's a quick starter question for you. Tomorrow I'm running a marathon and I'm not able to eat during the race, but I can drink. Now my friend has told me that if I drink pure water, I'm going to run faster, but my coach insists that this is actually quite dangerous. So I'd like you just to pause the video and to jot down whose advice you think I should follow and why. Okay, and so actually, I should really be listening to my coach. It is dangerous if I was to be doing a long period of exercise and only drinking pure water that didn't have anything dissolved in it, then what could happen is the quantity of dissolved ions and other sort of electrolytes in my blood would decrease, and this would actually affect the water potential of my blood, and it could affect it so much that my red blood cells might start to burst. So it's actually better for me to drink water with dissolved glucose and things like that in it, so that it doesn't affect my blood water potential. So using the progression line, I'd like you to jot down your confidence ratings for these 4 learning objectives. So for example, learning objective number 1, describe and explain what will happen to plant cells in hypotonic, isotonic and hypertonic solutions. Maybe following my independent study, I feel relatively confident on that. So I'm going to put that just up here and I'm just going to write a number 1 for that learning objective. Okay, so I'd like you just to jot down whereabouts you sit confidence wise for all of these learning objectives on this slide. Okay, so you might just want to pause the video and make a quick flashcard of this information. Now, when we're talking about water potential, if we're discussing pure water where there's no dissolved solutes in it, then the water potential value is always 0. And water potential can be represented using this symbol here, which is psi. So here we've got a water potential value of 0. Now we know that is pure water, nothing dissolved in it. If we start to dissolve some solutes in our water, which is our solvent, then our water potential value is going to start to become more negative. So here, maybe I'll dissolve some sodium chloride, some salt in my water, and my water potential value is going to move to -50. If I keep adding solutes, then my water potential value is going to get even more negative. So here, it's all the way down to -500. Now, the direction of osmosis is always down the water potential gradient. So here you can see that the water is going to move from an area of less to more negative water potential, and that's down the water potential gradient. So this is always the direction of osmosis. So I'd like you just to pause the video and to have a go at describing these 3 different solutions: a hypertonic, isotonic and hypotonic solution. You might want to use these images to help you. Now what these are is basically beakers with solution and then one massive cell in the middle. Okay, so here this represents a cell, and you can see that the water potentials of the solutions and also inside the cells have been represented. So please bear in mind these are tiny little beakers, and that's just one cell in each of those beakers. Okay, so a hypertonic solution. Now, hyper you might know of as meaning someone has lots of energy or more energy. So that might help you to remember that a hypertonic solution has got more dissolved solutes than the inside of the cell in this example. Okay, so for example here we can see that the water potential of the solution is -70, and we can see that the water potential of the cell is -10. Now, as we said on our last slide, water always moves by osmosis down the water potential gradient. So here, -10 is less negative than -70. So water is going to move down the water potential gradient from -10 to -70. And you might want to use your flashcards that we made on the last slide just to kind of help you monitor that. So water will move down the water potential gradient out of the cell into the solution. Okay, an isotonic solution. Now, iso basically means the same. So here we've got a water potential inside the cell that's the same as outside the cell. So there's going to be no net movement of water. There will still be water molecules that move in and out of the cell, but there's no net movement. Hypotonic, so hypo means fewer dissolved solutes. So here you can see that the water potential of the solution is minus 7, sorry, and that is not as negative as the water potential within the cell, which is minus 10. So in this situation here, water again is going to move down the water potential gradient from a less to a more negative water potential. So from minus 7 to minus 10, and in this case it's moving into the cell. So water moves down the water potential gradient into the cell. So if we look at that in terms of two different cell types, if we're looking at animal cells, you guys know that animal cells don't have a cell wall, they've just got a cell membrane. And the cell membrane isn't very strong, as you learned already. So the phospholipids, they're not chemically bonded to each other. They are attracted to each other and they form this phospholipid bilayer. So if you place an animal cell in a hypotonic solution, so this is a solution with fewer, because hypo means less, fewer dissolved solutes than inside the cell, water is going to start to want to move into that cell down the water potential gradient. And because the animal cell doesn't have a cell wall to provide structure, that cell is going to lyse or split. Okay, and you might remember that word lyse from hydrolysis, lysis meaning split. If you place the animal cell in an isotonic solution, it will just remain the same because you've got the same water potential inside and outside of that cell. If you place the animal cell in a hypertonic solution, again, remember that hyper means more. So we've got more dissolved solutes in the solution. Then water is going to want to move down the water potential gradient out of the cell, and your cell is going to become crenated or shriveled. Now, if we place plant cells in those same solutions, you're going to get a different result. And the reason for that is that a plant cell has this structural cell wall that stops the cell from lysing or crenating. But they do other things. So if you're going to place your plant cell in a hypotonic solution, then the cell is going to, or the cytoplasm is going to increase in volume and start to push against that cell wall. So you'll actually have some resistance from the cell wall pushing back on the cytoplasm, and that will eventually stop more water from entering into that cell. And in this state, this cell is called turgid. If you place your plant cell in an isotonic solution, then we can describe it as flaccid. And so again, there's no net movement in or out of this cell because the water potential inside the cell matches that outside of the cell. If you're going to place your plant cell in a hypertonic solution, then your cytoplasm of that plant cell is going to decrease in volume. So here, there's actually two things that are happening. Not only is the cytoplasm decreasing in volume, but also the vacuole. Now, what that means is that the cell is going to appear plasmolyzed, and you can see this because the cell membrane has pulled away from the cell wall. Okay, so you can see really clearly here that this cell is plasmolyzed. Okay, so we're going to have a go at activity 1, which is to sketch a cell comic strip. If you've got a surname beginning with letters A to J, I'd like you to draw 2 comic strips showing the effect of placing an animal cell in a hypotonic solution, and then another comic strip of what's going to happen if it's placed in a hypertonic solution. If you've got a surname starting with letters K to Z, I'd like you to draw a comic strip of a plant cell being placed first in a hypotonic solution, and then another comic strip of it being placed in a hypertonic solution. Once you've done this, I'd like you to take a photo of your comic strips and upload them to your team's channel. Have a look at everyone's on there and like which one you think is the most creative, but also scientifically accurate. Okay, so do get creative with this, and we've had some really fantastic cell comic strips in the past. So I'd like to pretend that I drew this, but I didn't. But here's a really nice cartoon representation of what happens to an animal cell when placed in those different solutions. Okay, I'd like you to have a go at activity 2, which is the water potential activity. So maybe just pause the video and have a go at that. And I'm going to talk you through my example now. So the first thing is I'm going to draw an animal cell, and this is my basic structure. So I haven't even put any organelles in it. And I need to make up a water potential for inside my cell. And kilopascals, as we remember, is the unit for water potential. So in my cell, I'm going to say that this has a water potential of -10 kilopascals. And just a reminder, you shouldn't have a positive number here because water is either pure and has a water potential value of 0, or it's got dissolved solutes in it. So then if it has dissolved solutes in it, it's a minus or a negative water potential value. Next, I'm going to make up a water potential for outside my cell, so the solution. So here I'm going to say that my solution has a water potential value of -700. Again, I'm going to make sure I put my units kilopascals. Next, I need to identify, is that solution hyper, hypo, or isotonic? So -700 is more negative than -10. And so I know that I've got more dissolved solutes in my bathing solution. So if I've got more dissolved solutes, hyper means more, and hypertonic is how I would describe that solution. Next, I need to draw an arrow to show the direction of water movement via osmosis. Now, as we know from that flashcard we made earlier, water always moves by osmosis down the water potential gradient, so from a less to a more negative water potential. So in this example, water is going to move from a less negative water potential inside the cell to a more negative water potential outside the cell. So water is going to move out of the cell by osmosis. So I'm going to draw some arrows on there. Write a sentence to explain why water moves in this direction. So again, water will move from a less negative to a more negative water potential down the water potential gradient. So that's one of our key phrases that we want to include there. So I'm just going to write that down. So down the water potential gradient. And finally, you should have taken a photo and uploaded your diagram to your team's channel. Do have a look at your other teammates and just see if they've made any mistakes and just let them know. It's really helpful to sort of spot any errors that others have made, and they can do the same for you. So we're going to talk a bit about exam technique now, and this is explaining water potential. So here our question is, a scientist placed an animal cell in a solution. 5 minutes later, she viewed the cells under the microscope and noticed that they looked crinkled. Can you explain what happens to the cells? So oftentimes people actually just give like 1-sentence answers for these, but there's a lot more key things that we can actually say when we're explaining the answer to this question. So here's our guidance, and it might be quite useful for you to just pause the video and to make a flashcard of this, because if you follow these bullet points, then often you can pick up very easy marks in these kinds of questions. So the first thing that we want to do is to say what kind of solution we're looking at in this question. So is it hyper, hypo, or isotonic? Does the solution have a more or less negative water potential than the cell? What direction does water move? So is it into or out of the cell? Name the process. That's really simple. That's just osmosis. But put that word in there because it's an easy one to forget. And then what happens to the cell? So is the cell lysing and splitting, or is it crenating? So let's look at the answer then. The solution is hypertonic. So it's got more dissolved solutes compared to the inside of the cell. Then, because the solution has more negative water potential than the cell, it causes water to move down the water potential gradient out of the cell by osmosis, leading to the cells shrinking or crenating. So if you just follow those key points, then you can very easily pick up marks in these kinds of questions. So next I'd like you to test your exam technique. So pause the video. You've got 5 minutes to have a go at this question. Try and write down 5 bullet points because it's a 5-mark question, and try and remember the guidance that we spoke about on the last slide. Okay, and the answer for that question So again, you might want to just pause the video and just check your answer, see if you can add any detail or if you've missed anything, and just add it in if you have. So as you guys know, placing plant and animal cells in the same solution, they behave differently, and that's because plant cells have got a strong cell wall that provides structural support to stop them lysing or splitting. So we need to be able to use this equation to calculate the water potential of a plant cell. And the water potential of a plant cell can be calculated by adding together the solute potential and the pressure potential. Now, the solute potential is the concentration of solutes in the cytoplasm and vacuole of that plant cell. Now, this is always a negative value because you'd never have like a pure water cytoplasm. You'd always have dissolved solutes in there for metabolic processes and things like that. The pressure potential is the hydrostatic pressure. So hydrostatic is basically water pressure from the cell contents pushing out on the cell wall. And this value is always 0 or positive. So here, the solute potential is going to be pulling water into the cell, but then the pressure potential is going to be pushing against that. And that's because the cytoplasm and the contents of the cell are pushing out on that cell wall. So let's try a few calculations on this. I'd like you to find the water potential when the pressure potential is 40 and the solute potential is minus 50. So hopefully you spotted that the water potential here of the plant cell is minus 10. Next one, can you work out the water potential when these are the pressure and solute potentials? And hopefully you spotted that it's minus 3,390. Next, try this calculation. So what is the pressure potential when the water potential is minus 200 and the solute potential is minus 250? So the first thing that we'll need to do here is to rearrange the equation. So we'll need to move the solute potential over to the other side, and that will give us this equation here where the pressure potential is equal to the water potential minus the solute potential. And we'll get a pressure potential of 50. So finally, I'd like you to try this question here. What is the water potential? And what we can see here is that the pressure potential, so the amount of pressure that the cytoplasm and vacuole are pushing out on that cell wall, is equal to the solute potential, which is pulling water into the cell. And we can see that they balance and our water potential is 0. So bearing that in mind, the cell is fully turgid when the solute potential is equal to the pressure potential. And this will always give a water potential of 0. Okay, please pause the video and complete activity 4. And this is all about graph skills. Okay, so the answers for this then. Question 1, using the graph, state the sucrose solution that is isotonic to the potato cells. So we should hopefully have noticed that there is no percentage change in mass when the sucrose solution is 0.25. How did we come to this conclusion? Well, at this value, there's no percentage change in mass of the potato cores. Question 3, what is the difference between plasmolysis and incipient plasmolysis? So plasmolysis is where most or all of the cell membrane will be pulled away from the cell wall. So here you can see that we've got full plasmolysis and most of that cell membrane has been pulled away from the cell wall. Whereas in incipient plasmolysis, the cell membrane has started to pull away. So here we can see incipient plasmolysis because that cell membrane is starting to pull away from the cell wall, but most of it hasn't completely detached, as you can see this picture here compared to that one. Please pause the video and complete activity 5. Now these are differentiated activities, so just spend 30 minutes, try to complete at least 2 of them, and it's up to you where you start. So if you're not feeling very confident about this topic, then you might want to start with a lower confidence activity and then work your way through. If you're feeling a bit more confident, then you might want to start with a higher confidence activity. And if you have finished those and you want extra, then we've got the extension activity as well. So if those of you had a go at the extension question, you might have already watched this video. If not, you might just be interested to have a look at it. It just talks about some of the adaptations that fish have for living in salt water. So there's a very cool water potential and osmosis rap video that you might want to watch that uses some of the key vocab that we've spoken about in today's session. And if you're not enjoying this pun, then maybe you'll enjoy this one. So finally, I'd like you to go back to regression lines that we did at the start and to reevaluate your learning objectives. So for example, for me, my learning objective 1, I think I said was about here at the start of the session. And now maybe after doing some of those activities, maybe my confidence has increased. So now maybe my learning objective is up here. If any of your learning objectives haven't moved and you're sort of feeling still not very confident on them, I'd like you just to take note of that. That's okay. Sometimes that does happen. And just write down some questions that you have. Maybe you can post these onto your channel on Teams, take them into lesson, or pop along to a subject extension. And do make sure you get answers for those questions, because it's going to help you progress much more if you're constantly asking and checking your understanding. Okay, thank you. Bye.